Good day everyone. Today we are going to start the in-text questions of chapter 5 and 6, that is molecular basis of inheritance and evolution. We have already completed the previous chapters and we have now come to the molecular basis and evolution. Moving on to the first question, that is on page number 82, that is why did space between two polynucleotide chains in DNA remains almost constant? So the answer is that the almost constant space between the two polynucleotide chains in DNA is maintained because purines and always pair with pyrimidines, so they create base pairs of a consistent size. So we all know that basically A will actually form bond with T and G will form bond with C. So it is easily understood that this is the constant pairing and therefore the size will remain equal throughout the DNA strand. Moving to next question, that is, in some viruses the flow of information is in the reversed direction, that is from RNA to deoxyribonucleic acid. So simple name to this process is reverse transcription. Moving to next question, that is, if the length of the E. coli DNA is 1.36 mm, then we have to calculate the base pairs in the E. coli. So basically we are given that the length is 1.36 mm, which is into 10 to the power minus 3 in meters, and we already know that between two consecutive base pairs we have 0.34 nanometers. Nanometers can be converted into meters with 10 to the power minus 9. So the number of base pairs can be calculated by the total length of the DNA by the space between two base pairs. So when we perform the calculation, we get the number of base pairs to be 4 into 10 to the power 6. Moving to next question, that is, theoretically, how many such beads do you imagine are present in a mammalian cell? That is, how many nucleosomes will be present in a mammalian cell? So after performing the calculation, we'll be able to get the answer to be 3.3 into 10 to the power 7. Next question, can you think of any difference between DNAs and DNA? On page number 85. So we all know that DNA is a genetic material carrying instructions for an organism's development and function, while DNA is an enzyme that breaks down DNA. Moving to next question, page number 87. That is, what are the two chemical differences between DNA and RNA? Sugar. So the basic differences we can find are in two components, that is sugar and nitrogenous bases. So in DNA we have deoxyribose, while in RNA we have ribose sugar, and there is an extra OH on the second carbon. Nitrogen bases in DNA we can find adenine, guanine and thymine, while in RNA we can find adenine and guanine are similar, but uracil is found instead of thymine. This is very important to note that thymine and uracil are the variations we can find in DNA and RNA. Moving to next question in page number 89, that is, can you recall what centrifugal force is? So we know that it is an apparent outward force experienced by an object moving in a circular path. So when an object is moving in circular path, it will experience an outward force which is called centrifugal force. The inner force is called centripetal, while the outer force is called centrifugal. The second question is that, think why a molecule with higher mass could sediment faster. So molecules with higher mass sediment faster in a centrifuge because they experience a greater centrifugal force, causing them to move towards the bottom of the tube more quickly. So if the mass is more, centrifugal force will be experienced more. They are directly proportional to each other. Moving to next question on page number 90, that is, if the E. coli is allowed to grow for 80 minutes, then what could be the proportion of light and hybrid DNA molecules? This combination is called hybrid, while this is called light, and heavy is also present, that this is called heavy. So this is hybrid, this is light, and this is heavy. So we know that each E. coli will take 20 minutes for its full growth and to divide. So when we calculate all this, we'll be getting the answer to be 14:2, and when we cross them, we'll get the answer to be 7:1. Moving on to next question on page number 92, that is, can you write the sequence of RNA transcribed from the above DNA? So we know that the upper strand will be the one which will be used as the coding one. Even though it will not code for anything, it is called as coding strand. So you have to take this as the reference. So when we take A, in RNA, we know that A will be not replaced by thymine, but it will be replaced by uracil. Then T will be with A, C with G, and we continue to do so, we'll be able to get the following strand of RNA. Moving to next question on page number 96, that is, we have to predict what is the amino acid coded by this sequence. So we know that AUG is a start codon which also codes for methionine. So when we check the checker box, then we'll be able to understand that UUU is for phenylalanine, and we can find UUC. So UUC will also code for the same thing, and UUC. So you can understand from the checkerboard that this will be the coded proteins. And then the next question here, that is, what will be the code for these proteins which are expressed? So for these proteins which are expressed, we can find the code like this, or we can change it to UUC, UUC, or some UUCs and some UUUs. Moving to the next question in page number 101, that is, can you think for how long the lactoperon could be expressed in the presence of lactose? So the answer to this question is as long as lactose is present. So as long as lactose is present, then that is, this will continue until lactose is present, that is, all the lactose is converted into glucose and galactose, then the reaction will stop. With this, we have completed molecular basis of inheritance. Now we'll move to evolution. The first question is that, do you remember how the ages of fossils are calculated? So the ages of fossils are calculated with a combination of relative and absolute dating methods. So the second question states that the method of radioactive dating and the principles behind this procedure. So we know that it's a combination of relative and absolute dating methods. So when we see the relative dating methods, it is that it compares the fossil to the known ages. So when we know some fossil about its age, then we can compare the new fossil which you have found with the previous one. This is called relative dating. When we see the absolute dating, that is, we will rely on the radioactive decay of isotopes to establish precise ages. Moving to the next question, that is, it is argued that if within hundreds of years man could create new breeds, could not have nature done the same over millions of years? Yes, it is able to do, but it is very selective in doing, because all the organisms should survive on the same planet. So if there are hundreds of them, it could be leading to an internal competition and nothing will be able to survive properly. So nature can do the same thing, but it's more selective in what it does. That's all. Thank you so much for watching. Stay tuned for more updates.